Hello, everyone, and welcome to today's Third Thursday webinar from the Michael J. Fox Foundation. I'm Dave Iverson, contributing editor at the Foundation, and we're delighted to have you join us for our final Third Thursday webinar of 2014, which fittingly is going to focus on the year in review in Parkinson's research, what's been accomplished, what some of the opportunities and challenges are now at year's end, and also then to talk about those in the year ahead. Before we get started, though, we also want to thank AstraZeneca for their sponsorship of this year's webinar series. And we also want to remind you that you can submit questions throughout the hour today. Just use the Q&A box, which is in the right corner of your screen, and then just type in your questions there, and then in the second half of our webinar, we'll do our best to answer as many of those questions as we can. And we also want to note that the slides that you're going to be seeing throughout the discussion today, you can download. So there's the resource list box that's also on the bottom of your screen, and you can click on that there. It'll open up in a different browser, and you'll be able to either save them or print them. We know that sometimes some of the boxes on the screen are sometimes a little bit harder to read, and you can move them around and resize them. So if you're having trouble seeing them, if you want to click on them and move them to a different location on their screen, make them bigger or smaller, you do have the option to do that. So let's get started and focus in on what we're going to be covering today. What we really want to do throughout our hour today is look at the year that's been and focus on a variety of things that have to do with our progress during the 2014 year. So we're going to talk some about what drugs are now being tested that we think may possibly play a role in halting the progression of Parkinson's disease and what advances have been made throughout the year. We're also going to talk, though, about advances that have been made in developing new treatments to treat various symptoms of Parkinson's disease, both motor symptoms and some of the non-motor symptoms as well. Then we're going to kind of step back and kind of look in a big picture way about what we've learned about the disease itself over the course of the year and how, of course, that will also inform more progress, and then talk finally about the role that all of us can play in speeding that much more progress in the year ahead. So let's meet who we're going to be having our conversation with today. Here are our panelists who are going to be joining us. Steve DeWitt, my colleague on the Michael J. Fox Foundation Patient Council. Steve's also been very active in a variety of Parkinson's efforts and organizations in his home state of Connecticut. Joining us as well will be Dave Weiner, Dr. Dave Weiner, who is the Chief Medical Officer at aTyr Pharma and also a member of the Michael J. Fox Foundation's Executive Scientific Advisory Board. Katie Koppel will be here, Dr. Katie Koppel, who is the Associate Director of Research Programs at the Foundation, and Dr. Todd Scherer, who is the Chief Executive Officer for the Michael J. Fox Foundation. So we're going to start actually in a little bit different way today before we plunge into our conversation. We're going to show you a little video, and this is a video that I actually narrated for the Foundation that describes kind of the research process, why it takes a while, but actually more importantly, the role that all of us can play in that research process and the need for research volunteers. So we want to play this short video. It lasts about a little over three and a half minutes, just as a way of giving us context for our discussion that will follow. So here's that video.
Hi, I'm Dave Iverson, contributing editor to the Michael J. Fox Foundation. I've been a journalist for 35 years, and I have Parkinson's disease. Today, I want to tell you a story that doesn't get reported often enough, the urgent need for research volunteers like you and me. I've participated in Parkinson's research, and maybe you have too, but fewer than 10% of Parkinson's patients make that choice, and a shortage of volunteers has become a huge obstacle, one that blocks progress and our path to a cure. Here's why. Developing new drugs is like running a long-distance race with a few hurdles thrown in for good measure. The good news is, it's a relay race. We're not running on our own. And for the first leg, we have a pretty big team. Government funders, academic scientists, research foundations, biotech, and pharmaceutical companies. This is where we take the aha moments around the first curve and into the middle stages of lab research. That initial stretch can take more than a decade and tens of millions of dollars. Even then, most new drug candidates never make it out of the lab. But a few, with the right stuff and sufficient funding, stay on course for the handoff to the next team of researchers, who then have to clear a tough but necessary hurdle: carefully designed human clinical trials. And this is where the relay team needs fresh legs, because for this part of the journey, the only racers who can grab the baton are people like us. Us means people who have Parkinson's, but also people who don't. Family members, friends, we're all needed to help complete the next stage of the race, which is called a Phase 1 trial. This is where a promising treatment is tested in a small number of people. If it's successful, we need a Phase 2 trial to determine preliminary effectiveness. And that takes, guess what, a larger pool of volunteers. If it makes it past that hurdle, even more volunteers are needed for Phase 3, which will determine if the drug is effective enough to warrant the pursuit of FDA approval. In addition, we need volunteers for so-called observational trials, where we track who's at risk and what it's like to live with Parkinson's. You get the picture. This race needs a really big team. And right now, we just don't have enough runners. The result is a dramatic slowdown in research progress. 85% of clinical trials face delays, and 30% never even get off the ground. The companies that pay for clinical trials know this too. Slow progress and delays are factored into their investment decisions, and often the dollars flow elsewhere. To find a way forward, we need to increase the roster of research volunteers now. We can't wait, and once you join, you'll find plenty of support and the right team at your side. Sure, the finish line will sometimes seem as distant as the sky, but as Michael J. Fox reminds us, cures aren't going to fall from the sky anyway. We have to get ladders and climb up to find them. Michael and his foundation are busy building the ladders needed to overcome scientific obstacles, but they can't do the climbing for us. We have to run this leg of the race ourselves and volunteer for research. And if we do, together we'll cross the finish line.
Okay, just a short video to give us a sense of some of what's involved in the research process and the way that we all can play a role in furthering that progress. And we wanted to give you a sense of that before we dive into what we're going to talk about next. And we're going to take up first some of the progress that's been made this past year in developing better therapies for dealing with the motor symptoms of Parkinson's disease. And Todd Scherer, why don't you start us out, if you would, CEO at the Foundation, about the first two items that we see on the list, because these are two of the items that are getting quite close to perhaps being available to people. And these have to do with ways that we can begin to contend with what's often difficult for many people living with Parkinson's, which is those ebbs and flows, the so-called on-off cycle, when your medication begins to wear off and you're not doing as well. Tell us something about these first two items, which are sometimes referred to as rescue drugs, ways that people could get sort of quick relief when they're beginning to experience that off syndrome.
Yeah, so we've, in the past year, there's been a lot of progress in trying to develop improvements in the ability to treat some of the motor symptoms of Parkinson's. And one of the areas that has gotten a lot of attention and made a lot of advances is really more around the better ways to deliver drugs that we already have or drugs that we know will already work. So, for example, the drug most commonly used for Parkinson's is Sinemet, which is levodopa-based therapy, and it's taken through an oral delivery. So just like with food that you eat or any other medication you take, when you eat something, it takes some time for the body to digest that material. It gets into your system, and then slowly your body gets rid of it. And because of that process, in the case of Sinemet, there's a period of time when the drug is in the system and effective, but there's also a period of time when you're waiting for the drug to kick in. And that can result in and off the fluctuations, the on and then the off. So the on is when the drug is in the system and working. Then the off is when there's not enough drug in your system. And normally what the patient would then do is take another pill and try to get more of the drug on board. But there's a delay. There's a period of time while the body has to process that oral drug. What these first two drug therapy approaches are focused on are developing more rapid ways to get the drugs into the system. In one case, the case of Accorda, it's to try to develop a better way to get levodopa into the system more quickly. And in that case, they're developing it through an inhalation delivery. So think of an asthma inhaler where they've aerosolized levodopa so that it can get into the system very rapidly through the lungs. And in Synapsis, they're working on a drug that's called apomorphine, which is normally given right now through an injection. And in the case of Synapsis, they have reformulated that medication so that it could be given in a delivery what's called sublingual. Sublingual is if you think about a Listerine strip. So both of these have advanced very quickly. They can get the drug on board very rapidly to overcome an off symptom and are in late-stage clinical testing.
And David Weiner, why don't you pick up the story from there and describe a little bit about the next two things that we see on the screen, which are, again, ways to alleviate that on-off problem, but rather than in the kind of instant rescue way that Todd was describing on the first two, these are more about providing levodopa in a more continuous way, either through by way of a patch or an actual intestinal gel.
Yep. So as Dr. Scheer was stating, L-DOPA, levodopa, is a profoundly effective medication for Parkinson's disease. But like all medications, it has some drawbacks or some negative attributes. And one of the significant ones with levodopa is over time, as you take that medication over a period of a few years, you can develop complications like extra or adventitial movements like dyskinesias, in addition to some of the fluctuations that we just talked about with the other two medications. And one of the dominant approaches to try to minimize the development of those complications is to try to provide a more even exposure or more even levels of L-DOPA in the blood during L-DOPA therapy. So as Dr. Scherer mentioned, Sinemet, and the way your body handles that is you usually have a fairly high level that drops off fairly rapidly. These two approaches here are utilizing either bypassing the intestinal tract or using a through-the-skin delivery to try to approach this so that the levels are much smoother throughout the day. And the anticipation is that that will minimize or reduce the development or the propensity to development some of these dyskinesias and fluctuations. And we do have significant data that is very promising in that regard, that these approaches are going to lead to an improved way to manage Parkinson's disease with the use of levodopa.
And David Weiner, we saw in the video what a long process this is, and I don't think it can be emphasized enough how close we really are here. We see at the top bar everything from, the beginning process where you're first trying to figure out what the right drug target is all the way to phase 3, and we see how long that takes and how you have to kind of worry your way through that in the research process and kind of place your bets and hope that it all proves right. It's a long, long process, and what's exciting now is that these things are really close to the finish line.
Yes. This is a reflection of the renewed interest in Parkinson's disease as a disease that we could develop very effective therapies for. The foundation has played a role in that. A lot of what you see now is bearing fruit from activities that occurred years in the past, and I think that optimism moves forward. So a lot of the activity that's being done in this community, both with the foundation, with small biotechnology companies, as well as some of the larger pharmaceutical companies, there is a significantly renewed interest in Parkinson's disease, in part because we're improving our understanding of the disease. And there's technologies that are being developed. Some of the things on this particular slide revolved around technologies that were not specifically developed for Parkinson's disease, but were being applied to that disease because of the advantage it could have in managing patient symptoms and ultimately slowing down or arresting the progression of the disease. So I couldn't agree more that the limiting factor now is really the availability of participants in clinical trials to match up with the exciting science and the advancements that are going on across multiple stakeholders in the development of new treatments for Parkinson's disease.
And Steve DeWitt, we'll get a little bit later into more of the things that patients can do, and you've played such a leading and innovative role in fostering that in your state of Connecticut. But I'd also like you at this point to speak to the role that patients can play in sort of making the case for why this is important. You've actually done some work in this area, talked to insurance companies and others about why it's important to cover some of these new medications, because it can seem at first glance like the on-off cycle is kind of an inconvenience. Oh, well, my meds aren't working quite as well right now, but they'll work in time. It doesn't seem like that big a deal. But you have, I know, can make this point in a really direct way, so I'd like to ask you to do that, Steve, about why, for example, the on-off cycle is not just an inconvenience, and why it's important for patients to play not only a role in volunteering for research, but also for advocating for why that's so important.
Thank you, Dave. I was privileged to be part of the payer meeting that Fox Foundation held with insurance providers and Social Security benefits groups, and they held to the belief that there was limited need for this kind of medications. And I point out two examples that I've seen within my support group alone, in which patients are concerned about being behind the wheel at a time that they might go off and looking for some kind of relief, whether it be having to pull off the side of the road medications or something like an inhaler that could help for the moment to just get them to safety. Another example was a gentleman I know that is an electrician who had a freezing episode while he was working. Definitely moments that hit hard with the payers, seeing the value of these kind of medications that formerly they just thought was a matter of convenience.
And Todd shared that making that case is important for a number of reasons, not only to convince insurance companies that these medications ought to be covered, but also to really make the point in the final stages of this, right, with FDA approval, that these aren't just small steps. These are things that can really make a difference in the lives of patients.
That's right, and it's also important so that the companies that are working in these areas are willing to put the investment in to get the therapies all the way through the clinic. I mean, one thing that should be pointed out when you look at these slides is that it gets exceptionally more expensive each step along the way in doing these larger trials. So what we are trying to do is make it clear to all the players in this field, the biotech companies, the funders of the research, the regulatory players, both on approval of the drug and then payment of the drug, to make it clear to them that these are very clear unmet needs for patients. We have potential therapies that could address those needs, and we need to go full force forward to push those therapies forward as quickly as we can to get them into the hands of as many patients as possible.
And before we move on to our next slide, which is to take a look at some of the progress we've made in treating non-motor, there are a couple of questions coming in right now about some of the therapies that are listed on this slide. So let's go ahead and just address those right away. Dave Weiner, maybe you can take these. A couple of people are asking about the last one in particular, this intestinal gel. One question is, how's that gel delivered? And the other question is whether or not, if you use that gel, whether or not then that has an impact on the rest of your diet, if it's something that's being delivered directly into the intestinal system.
Happy to. So this approach involves a surgical procedure where a tube is placed across your abdomen and into your duodenum. So this is a part of your intestinal tract a little bit further down from your stomach. And the intent there is to deliver levodopa directly to the area where it is most efficiently absorbed into your system. And that gets around a lot of the problems that many Parkinson's patients have with the emptying problems that occur in the stomach. So this is a therapy that is not appropriate for all patients because it has an invasive component to it. But for a particular subset of patients that have particularly severe fluctuations that we were talking about, this can be a profoundly effective therapy to help smooth those out. Okay? This is actually already approved in Europe and is now getting very close, as it states, to a potential approval and more widespread use in the United States. And the following question regarding diet, I'm not aware of whether this impacts or changes your diet in any case, meaning that you can continue to have your usual diet even once you have this therapy administered.
And Dave Weiner, I think I probably didn't quite grasp initially, and I think now I do, the question from Kevin, which I didn't really state very accurately, which I think what Kevin is really getting at is, there's sometimes a problem with the absorption of levodopa and protein. Sometimes if you have a big protein meal, that gets in the way of the way in which levodopa is absorbed into your system when you're just taking the normal oral Sinemet. I think Kevin is wondering whether or not this gets around that problem.
Yeah, it helps to address that problem. I'm not certain that still a profoundly high protein meal, whether that would affect the effectiveness of the duodopa intestinal gel, but the whole principle behind delivering the medication at that part in your gastrointestinal tract helps to alleviate some of those interferences.
So suffice to say, in this arena, really significant progress, getting very close, so that within, as is footnoted at the bottom of the slide, within the next year or two, many of these new approaches, which could make a real difference in relieving some of those on-off problems that Steve DeWitt was describing, will be available to patients, we think, very soon. So 2014, a real year of progress in terms of better treatment, getting closer to better treatment for motor symptoms. Let's move on to our next slide, which has to do with what sort of progress was made in an area that's in some ways I think more challenging, which is progress in treating some of the non-motor symptoms of Parkinson's. It's really only been more recently that we've begun to focus more on these issues that are a part of Parkinson's and which in many ways, as we've talked about in webinars past, can be as debilitating or problematic as the motor symptoms themselves. So touch here again on the first one we see, which is a concern, of course, that everyone has, which has to do with cognitive impairment. Bring us up to date on the progress with this particular drug from Biotie.
Yeah, so I think that you framed this very well in that there's a huge effort now, an emerging effort, to really develop treatments for the non-motor symptoms of Parkinson's, which for the most part don't have very good treatments and are untreated. And in this area, we're starting to see some very promising prospects. And I think the most important step that we've seen in the last 2 years is a much more active portfolio of drugs being tested in the clinic. And in this case, from Biotie, they are developing a treatment that could reduce some of the cognitive symptoms that are seen in Parkinson's. particularly the early cognitive changes or more subtle, mild cognitive changes that are part of the disease. And what they're trying to do is target some of the other brain systems and neurochemicals in the brain that are affected in Parkinson's beyond dopamine through this SYN120 medication. And this trial was just launched in the last year. It's a phase 2 trial, so it is looking at efficacy and is now actively ongoing.
And Todd, while you have the floor, why don't you go ahead and take the last one, which is the drug for constipation, which again is one of those problems that never great fun to talk about, but can be really problematic for people with Parkinson's, and a new drug that's on the way that could be helpful in that regard.
Yeah, I mean, this is another symptom that is becoming more appreciated by the medical community, although obviously the patients have known about it much longer, and it gets to that point about the involvement and interaction of patients in the research effort to make sure that the priorities of the unmet needs are being focused on in the research. In this case, this is a medication that was being developed completely outside the scope of Parkinson's for digestive problems and other diseases. And in this case, the foundation is working with some Parkinson's researchers to try to reapply or repurpose this medication for work in Parkinson's. This trial also is just underway, so this is one of the first real multi-center trials looking at constipation in Parkinson's disease. So we're very interested and enthusiastic about the trial and also about the future of trying to develop treatments for this important symptom.
And Dave Weiner, take up the middle one, if you would, on describe first, if you would, what Parkinson's psychosis means, and then what the drug is that's being discussed here, because this was a drug that you were involved in some of the earlier research work on this before it got all the way to phase 3 and that much closer to being available for people's usage.
Mm-hmm. So as part of the spectrum of problems that can occur that are not directly related to motor function and more closely related to cognition, psychosis can occur in a number of Parkinson's patients, and it involves the development of hallucinations. Many of them are visual, where visual appearances occur in patients that don't necessarily actually exist. Some of them could be auditory, and those occur much less frequently in Parkinson's patients than some other types of disorders that have this term psychosis. Many people may appreciate disorders like schizophrenia and related disorders where psychosis is a major prominent component of those disorders. And the problem in Parkinson's is that the existing medications that are used to treat patients with some of these other disorders like schizophrenia block the dopaminergic system. So they could not be used in Parkinson's patients. So the initial challenge in trying to develop a drug for this was to come up with a fundamentally new way to try to treat these hallucinations and delusions without blocking the dopaminergic system. And when we were involved in the early development of this therapy, one of our principal goals was to make sure that it did not worsen motor function in Parkinson's patients. And not only does it not worsen that, but actually has, as you mentioned, Dave, has gone on to show profound efficacy in phase 3 studies, has recently been designated as a breakthrough therapy in the United States. And my understanding is that the process to try to apply for marketing approval is ongoing and could occur as early as next year. So this represents the culmination of a significant amount of work for a truly fundamentally new therapy for Parkinson's patients.
And before we move on to our next slide and get into really the heart of the research mission, which is to figure out a way to halt and slow down disease progression, just want to ask a kind of bigger question about the non-motor side of this, because as Todd Scherer was suggesting, patients have known for a long time that Parkinson's is highly problematic in ways that go beyond motor symptoms. It's now beginning to get more attention within the medical and research community. Katie Koppel, from your standpoint as Associate Director of Research Programs at the Foundation, is this something that you and your colleagues are spending more time looking at as you sort of survey the research world and look at the kinds of research projects that are out there that you think are promising and might be deserving of foundation support? Is more and more attention going into the non-motor side of the disease?
Dave, that's a great question, and I think from our funding perspective, we have seen an increase in the number of applications for researchers trying to develop new treatments to address the non-motor symptoms. And I think not only does that reflect the state of the field, but it reflects a better understanding of what Parkinson's disease is. And we sort of think of Parkinson's disease as a snowflake. There are lots of different types of Parkinson's, and one person's might not be the same as someone else's. And so as the motor symptoms of the disease are better managed by some of the symptomatic therapies that are in development, we can see more of the non-motor symptoms coming to light, and that has helped drive a lot of the research interest. There's a better understanding of the fact that these areas exist, and consequently there's more research that's coming online to help address some of these non-motor symptoms.
And Steve DeWitt, if you would add on to that from a patient perspective, how would you see it as important for patients to play a role? We saw in the video earlier that we're all needed to participate in research. But say something more, if you would, about the advocacy role that patients can play, even if it's just with their own doctor, and making clear the importance of these non-motor symptoms and the need for more research to be directed in that area.
Thanks, Dave. I would agree. The communications we have with our doctors is very important, so they can relay it back to the research community what they're seeing directly in the field. But I'd also make mention, too, that when you take a look at the medications listed here, over half of them have taken positions of being involved with the patients in the development of their protocols. Independent of being asked to, they do it voluntarily. So I feel the patients are active right now with some of the work that's being done, and I commend Fox Foundation for recognizing those kind of partnerships with companies that have the patient involved at the very beginning of the process.
Thank you. Let's push on then and begin to talk about sort of the central focus, of course, of all Parkinson's research right now, which is not only to do a better job of treating symptoms, managing the kind of day-to-day life of the disease, and we've seen a lot of progress being made in that arena in 2014. But now let's drill down on progress in terms of stopping or slowing the disease progression itself. Toucher, last month on our Thursday webinar, we focused on the problem of alpha-synuclein, that sticky protein that clumps up in the brain and that is toxic to cells, and that so much energy now is being focused on ways in which we can rid the brain and the body of those clumps, because we think that's key to stopping disease progression. And we see on the slide here two vaccine approaches against alpha-synuclein that are advancing now in phase 1 and in soon, we hope, into the next phases of progression. Describe what that vaccine approach is and how much progress was made in 2014 into getting this further towards the clinic.
Yeah, so I think one of the key features for where the next phase of progress is going to happen in trying to develop disease-modifying treatments really comes from a point that Dave Weiner made earlier, that we really are increasing our understanding of what may cause Parkinson's and how it may progress. And this protein, as you've mentioned, alpha-synuclein, has risen to the top as a key feature of the disease. What both of these approaches are trying to do is to figure out ways to get the body to break down or reduce the amount of alpha-synuclein that's in the brain. And we have a hypothesis that the increased amount of alpha-synuclein in the brain might be what's responsible for the disease and the progression of the disease. Both of these, the Epheris study and the Prothena study, were the first trials that ever went into people to try to target alpha-synuclein. So this is really uncharted territory and real innovation. And what's the most important and exciting result is that so far these have been safe. That was the first thing that you would focus on when you go down a completely uncharted path. And the next phases of these will be to really determine whether we're having any benefit with these therapies for people with Parkinson's. It's really, I think, these are the cutting edge, the leading edge of therapies that are targeting this particular. Know that there are many following along behind them with the real potential of slowing the disease.
Yeah, it's worth noting that while in 2014 the vaccine approach is the one that's sort of advancing first along these lines, there are a number of others that are sort of, if we go back to that relay race metaphor that's used in the video, there are a number of others kind of ready to start from the starting blocks and get into this two different ways of attacking alpha-synuclein, right, Todd? So even if the vaccine in the end isn't the thing that we hope it to be, and it may well be, it's not the only runner that we have in the race, right? I mean, there are a number of other approaches that we hope will be pushing along in 2015 and 2016 that will also attack alpha-synuclein.
Yeah, that's exactly correct. And I think the other important point is that there's been work on alpha-synuclein for many years. These two trials sit on the building blocks of a lot of research worldwide. But we really now have hit that critical inflection point of transitioning from the theoretical to the potential practical by having real potential therapies. And while, the first one may not be the most successful, they certainly will be the most, so that we will really understand how to go about testing these therapies and give us the best chance for success in the future if these themselves are not successful.
And we've got a couple of other points, and then we're going to also start taking your questions in just a moment. So do go ahead and add your questions in. We'll get to those in just a few minutes. But we want to mention a couple of points here in terms of ways in which we think there may be some opportunities to slow down the progression of the disease itself. David Weiner, if you would, tackle these two. One of calcium channel blockers. It's actually a blood pressure medication that we've discovered through observation that people who are on it appear to be less likely to get Parkinson's. Describe, if you would, what's going on with that particular approach.
So this is born off of some fundamental observations regarding the electrical activity of the brain. So the brain is an electrically active organ. It's one of the major ways that cells communicate with each other. And through basic research on understanding some of that electrophysiology in key regions in the brain, a hypothesis was generated that suggested that blocking a certain type of what we call a channel or a protein that sits in the membrane of a cell that helps move some of these electrically active ions across cell membranes could be beneficial in Parkinson's disease. That the abnormalities there help to drive some of the cellular death in certain regions of the brain. And it just so happens that existing medications, and you mentioned this, a certain class of antihypertensive medications, can block these and other channels. So what we often call a repositioning, or taking, in this case, an existing approved drug and trying to see if it could be repositioned or approached for the treatment of Parkinson's disease, not only to test this hypothesis, but to see if we can slow the disease progression. And that approach is now just about to reach what we would probably call the phase 3 stage of clinical testing, meaning broader patient populations to look for efficacy as well as safety of what was originally a hypertensive drug, but could potentially slow the progression of Parkinson's.
And last on the list, David, take this one too, if you would. Or it says neurotrophic factor optimized delivery. Growth factors, as probably lots of people are familiar, have long been hoped for as a way of slowing down disease progression. These are these proteins that boost and support dopamine neurons in the brain. But and the idea has been if we could increase growth factors, that would support dopamine production in the brain. But it's always been tricky figuring out how to make that happen. And some of the highly touted, highly anticipated efforts to make growth factors appear in the brain have not been successful in the past. But they're not dead yet, David Weiner? I mean, our growth factors are still a possibility?
Yep. So as you mentioned, these groups and their proteins are fairly large molecules, are amongst the most potent. They have the most dramatic effects when we test them in animal models of the disease. And the initial attempts over the last roughly 10 years or so to try to translate that into human therapy have run into difficulties, most predominantly with something we call delivery, meaning trying to get enough of these proteins to the right regions of the brain for them to be beneficial in Parkinson's disease. And the development of new therapies is an iterative process. So although we were all disappointed that some of the prior efforts did not succeed, we've learned something from each and every one of them. And so many of these growth factor therapies are now being optimized with respect to the way that they are being delivered to the brain. And obviously this is one of those ongoing trials, and there are others either in place or being planned to try to continue to exploit the profound biological effects of these molecules and fine-tune it in such a way that they can be administered safely and effectively to Parkinson's patients.
And David Weiner, a question coming in while we're on this slide, and then we're going to move on to our next one, but let's take this question now. A question about whether or not this calcium channel blocker would have an effect on either heart rate or osteoporosis. This is a medication that's, as you say, has already been approved for treating hypertension, so this individual is wondering whether or not it could affect either osteoporosis, bone density, often a problem in people with Parkinson's too, or heart rate.
Yep. So that's a very important question, because when you reposition a drug, you don't necessarily remove or change what its underlying other activities are, right? So in this case, one of the big challenges in the intermediate stages of evaluating this was what the effects would be on predominantly blood pressure, less so heart rate, but very much blood pressure. And the challenge in this is can we administer or give a large enough dose of this medication so that we can get effects in the brain without profoundly lowering patients' blood pressure. And that is one of the key outcomes or the key challenges as the drug moves forward. I can't necessarily comment on osteoporosis, so rather than stating something, I'm not really sure about the link there.
Okay. Thanks for raising the question, and David for providing the answer. Let's move on to our next slide, and as we do so, we'll now start folding in the rest of your questions at all, because at this point in our conversation, we sort of want to step back and sort of take a bigger picture look on all of the things that we've learned about the disease this past year, and as we go through this discussion, we'll add in your questions as well. And to get us started on this, Katie Koppel, this is really your focus within the foundation, is looking at one of the key things that's central to making progress in research, which is to be able to identify what's known as a biomarker, an actual precise way of both detecting Parkinson's early on, diagnosing it, but also measuring what's going on, because we don't right now, beyond clinical observation, as people who have participated in these webinar discussions before know, we don't have a very good way. We don't have very good yardsticks for actually measuring the disease. Some real steps forward in 2014 in that regard. So take us through this first point about we're now being able, we've heard a little bit about alpha-synuclein already, but one of the key things, since it's our culprit, it's the thing we want to get after, is we need to be able to measure it, right? So we're learning some things. We've learned some things this year. What did we learn?
Yeah, Dave, this is something that I think is really exciting about the outcomes of 2014 is progress made in biomarkers, and these really are the rulers for success when you're measuring whether a drug has an effect or not. And so, thinking about biomarkers, they could be many things. They could be heart rate or high blood pressure, telling you about cardiovascular disease. It could tell you just sort of scales about how much a disease has progressed, but in this case, we're really thinking about biologic advances. And so what we have learned from the Parkinson's Progression Marker Initiative study, which is a flagship longitudinal study that tracks people with Parkinson's disease and without Parkinson's disease over time, is that the protein, alpha-synuclein, that clumps up in the brain of people with Parkinson's is actually a lot lower in the spinal fluid of people with Parkinson's compared to healthy control individuals. And we think that alpha-synuclein is lower in the spinal fluid because it is clumping up in the brain and it's not getting into the spinal fluid. And so this is one of the first major findings of PPMI, that you can see this difference in PD individuals and controls over a lot of different centers, a lot of different countries. And this will be an informative tool for drug developers that are trying to develop therapies for alpha-synuclein in particular. And another exciting highlight is the idea that we could look in blood for the same marker. So alpha-synuclein in CSF is great, but it also requires a spinal tap. And so wouldn't it be wonderful if you could look at the same thing in blood when you go to a doctor's office and it's just a normal needle stick? And there was a researcher at Harvard that looked at blood samples from PPMI, and found that a precursor to the alpha-synuclein protein, something called RNA, is actually also decreased in people with Parkinson's disease compared to the healthy controls. And so this is another exciting advance that will inform better diagnosis and better clinical trial development.
And so, Katie, is the significance of this not only diagnostic, but also in time what we need to be able to do to figure out whether or not that vaccine that Todd was describing earlier, a vaccine against alpha-synuclein, actually works, is some way of detecting whether it works, right? So would one of these, being able to measure it better in the blood, or I know you're also working with other researchers to develop ways of imaging alpha-synuclein in the brain. I mean, we need these not only for diagnostic purposes, but also to know whether or not this big effort to defeat alpha-synuclein is actually working.
That's exactly right. And biomarkers are probably the most critical tool that we need to give drug developers to help clinical trials advance. And one example of how these results, the alpha-synuclein protein levels in spinal fluid or the precursor levels in blood, are contributing to that, or companies can look at the data from PPMI and then understand whether or not these markers would be good tools for drug development. So an example, one new finding this year is that we see in early Parkinson's disease the levels of synuclein protein in the spinal fluid are very stable over time. And so drug developers can then use that, knowing that over the course of a year, let's say, you wouldn't normally expect to see a change in synuclein levels in spinal fluid. If their drug changes those levels one way or another, they know that the drug is having an effect, and that increases their interest and their confidence in moving a particular drug farther down this relay race.
Let's fold in more of our questions now. I know there are several more bullet points at the slide. We'll try to get to those as we can, but we've been covering a lot of material in this hour, and that's meant that I haven't done as well as I would like in terms of answering questions from our participants. So let's start folding those in, and we'll try to get in more of our information as we can. A number of people are raising questions, Todd Scherer, about our old friend stem cells, which, in years past would have been perhaps more of our focus on this kind of conversation about research progress, the role of stem cells. What happened with stem cells in 2014?
So stem cells are still a very important and active area of research. There's really two directions that we're taking stem cell research in for Parkinson's. One, I think, is the one that people really think about, which is related to transplants. So taking stem cells, making them into, in this case for Parkinson's, the dopamine-producing cells, then transplanting those cells back into the brain in order to try to replace cells that had been lost in the disease. We have, over the years, become very successful in making dopamine cells in culture, in the laboratory, out of stem cells, particularly embryonic stem cells. But we've had a lot of challenges in trying to transplant those cells and get them to survive and continue to produce the dopamine that we want and have the characteristics that we want when put into the brain. So that has been a major technological roadblock. It's also, I think, important to point out that stem cell therapy in this context would really be another way of improving the delivery of levodopa. Basically, the stem cell would become a dopamine pump or dopamine sink in the brain. So we do have other areas that are further ahead of improving, getting more continuous delivery of dopamine for Parkinson's patients that we've talked about. The other major direction of stem cell research that has advanced quite aggressively in the last few years is really around using stem cells as models to better study disease and test potential new medications. So a major advance came from the ability to take a cell from an adult, so a skin cell, for example, and convert that back into a stem cell. So basically, you could then create the disease in a dish. So take a skin cell from someone with Parkinson's, convert that back into a stem cell, and then make it into a brain cell, and then study what's happened in that cell that might be different from somebody who doesn't have Parkinson's, and use that to increase our understanding of the disease as well as test potential medications to correct whatever the abnormality is in that cell. That area has advanced significantly in the last few years. Sorry, Dave.
Yeah, sorry to interrupt, Todd. I just wanted to sort of draw attention to the second bullet point on this slide where we see the word LRRK2, LRRK2, which is one of the genetic areas which can lead to Parkinson's. A mutation in the LRRK2 gene can lead to Parkinson's. This is where that stem cell technology that you're talking about can play a role, right? That we could take someone who has the LRRK2 form of Parkinson's, take one of their skin cells, turn it back into a stem cell, see how that disease then unfolds in a petri dish, and try to figure out a way to solve that problem? Is that sort of where this could go?
Yeah, that's exactly right. So in neurological diseases like Parkinson's, we are challenged in that we can't actually view or study very well the organ that is most affected in the disease, which is the brain. So if you compare it to, for example, in cancer, they can remove the tumor, they can study and analyze the cells that made up that tumor in the laboratory to really understand what went wrong that led to the disease. So we need to be creative and innovative in Parkinson's, and this use of stem cells gets us as close as we can to basically trying to understand what could be happening inside the brain of someone with Parkinson's, because we could make brain cells from a stem cell that came from that person and then really try to correct what might have gone wrong. So it has great, I think, hope and opportunity to really understand what might be underlying the disease and then develop interventions to prevent or reverse that. But this is definitely more in the future.
Let's get to more of our questions, and as we do that, let's also advance to our last slide and fold into the final part of our conversation, the ways in which people can get more involved in speeding research. We do want to draw your attention to that. As we mentioned in the video earlier, all of this research that we've been talking about throughout the hour can really only advance if people get engaged in clinical trials. And one way to do that is registering, as you see on this slide, at foxtrialfinder.org. A number of questions, Dave Weiner, coming up in about deep brain stimulation and its connection to research. And I want to hear more from Steve DeWitt about how people can participate in research. But one question that people ask is, if I've had DBS, can I still participate in research?
Well, that question depends very much on the individual clinical trial. For some therapies in development, having had DBS previously could create a confound or a problem in trying to understand or meet the goals of that trial. And usually in that trial, patients who have had DBS may be excluded. But a significant number of trials do not necessarily exclude patients with DBS. So the feedback is very much so regarding it's a trial-by-trial basis. And if you've had DBS and you're interested in participating in a trial, tools like the FoxFinder, like clinicaltrials.gov, and speaking about this with your doctor or movement disorder specialist can help you navigate which trials you could be eligible for.
Dave, just to also add, so I know we're trying to move on, but there actually are still studies going on within Fox Trial Finder, they're listed, that are trying to better understand the impact of DBS itself. So people who have had DBS, there still aren't a great number of people who have gone through that procedure, and there's a lot of active research to really still try to understand particularly the longer-term benefits and impacts of that therapy. So even that type of observational study, taking advantage of a treatment you may have already had, could be incredibly valuable to provide more information to people in the future who are considering that particular therapy.
And Todd or Katie, there are also a number of people who are asking about new advances in DBS, again, deep brain stimulation, which historically has been particularly effective at dealing with some of the motor symptoms of Parkinson's, tremor in particular. Are there new things being looked at in ways in which DBS might be effective for other symptoms of Parkinson's, other motor symptoms such as balance, perhaps non-motor symptoms as well? What's the latest on DBS research? Todd or Katie?
So there actually there's, I would say, two main directions also of this research, and we had some funding initiatives on this in the past year. One direction is actually to try to improve the surgical procedure and the technology that relates to the current application of DBS, using either smarter electrical devices or more fine-tuned devices or different ways of doing the surgical procedure in order to reduce the risks or complications associated with the actual surgery. So that's one area of research that's going on. The other area is the one that you referred to, which is trying to see if by targeting other regions of the brain or other stimulation parameters, if we're able to treat a broader scope of the symptoms that are impacted in the disease. And both of those are pretty active areas of research.
And Steve DeWitt, let's return to the subject of patient involvement. A couple of people are asking the question, how can I participate if the studies are not in my area? We see one of the bullet points on this slide, which is carpool, exclamation point, which is you're sort of the king of carpools in Connecticut in terms of getting people to various clinical trials when they might not be next door. But talk a little bit about you, if you would, Steve, about the way in which you've engaged people in Connecticut and the way in which you can get involved in Parkinson's research, even if that research project might not necessarily be very nearby.
I'd like to say first that the patients are extremely interested in getting into the trials, and sometimes the stumbling block is just getting from point A to point B. And so what we did in Connecticut was set up a transportation program to the major research centers in Boston and New York, and they're taking them the 2 to 3 hours it would take to get to the site so they can get to those trials. There's so much reason to be optimistic about what's been covered today, about the different treatments that are about to come out of the funnel. But my fear is that we'll forget that there's still a need to get many of these studies past their phase 3 period. And that means that we need to get enrolled in them, contact the research centers through Fox Trial Finder or directly, and get these to the finish line.
And Steve, last word from you on sort of what you'd also like to encourage on the medical side of this, ways in which the medical community, the research community, can encourage people to make, I guess, the research experience for volunteers more user-friendly.
I think the different companies that I've worked with in developing protocols for a friendlier experience, when they just listen to a patient talk about what it takes to get up in the morning, take their medications, and get through their day, they build those kinds of considerations into their protocols so that it's much more easier for people to get involved with the studies. And so it comes down to just encouraging them to ask the questions, and certainly the patients make sure they're available to respond and support some of those changes, because it'll just get the studies done more quickly and more efficiently, more cost effectively, and get us past this disease.
Steve, thank you. Thanks for all that you do in Connecticut and elsewhere. It really is true that we can't make more progress unless people participate, and you've done as much as anyone I know to encourage that among the patient community. I also really want to thank Drs. David Weiner, Todd Scherer, and Katie Koppel for not only their participation in this webinar, but also for all of the work that they're doing to further our overall goal. I think it's fair to say that 2014 has really been a year of significant progress, one that sets us up for, I think, more progress in the year ahead, progress that can happen most readily. If you do decide to participate in that research, you see the ways in which you can do that on the screen now, and we encourage you to consider that perhaps as a New Year's resolution for 2015. With that, we'll bring our webinar to a conclusion. I'm Dave Iverson at the Michael J. Fox Foundation, wishing you all the very happiest of holidays and happiest of New Year's. Thanks for joining us.